The complete guide to the phage display technology. Background of phage display technology. On October 30, 2018, the Royal Swedish Academy of Sciences announced that half of the 2018 Nobel Prize in Chemistry would be awarded to American scientist G. Smith and British scientist Sir G. Winter in recognition of their contribution to the invention and application of phage display technology. 1. Development of phage display technology. Literally speaking, bacteriophages refer to viruses that can eat bacteria, i.e. a group of viruses that can infect bacteria, fungi, actinomycetes and spirochetes that are very small in size with morphology varied from species to species. Generally, bacteriophages can be tadpole-shaped, small spherical or slender rod-shaped. Most of the bacteriophages are tailed dihedral structures, while some are not. The capsid proteins regularly arrange to form the head of the icosahedron containing the genetic material, nucleic acid. The filamentous bacteriophage is linear in shape and has no obvious head structure. Its capsid protein particles form a spiral structure, which encapsulates the nucleic acid of the bacteriophage. At present, according to the phage proteomic tree taxonomy, bacteriophages can be classified into 8 categories: smooth bacteriophage, cystic bacteriophage, filamentous bacteriophage, microphage, short-tail bacteriophage, long-tail bacteriophage, muscle-tail bacteriophage and fusilloviridae. Frederick William Twort, a British microbiologist, firstly discovered the phenomenon that bacteriophage can kill bacteria in the culture of Staphylococcus aureus in 1915. As a natural enemy of bacteria, bacteriophages are quickly adopted in the treatment of bacterial infections. In 1919, bacteriophage therapy successfully cured childhood dysentery in France. Later, it was found that bacteriophages can infect many pathogenic bacteria, such as Vibrio cholerae and Bacillus anthracis, and can lyse these host pathogenic bacteria. It has become a specific drug for the treatment of these pathogenic bacteria infection-related diseases and one of the effective methods for anti-infection treatment. With the invention and use of antibiotics, the use of bacteriophages for the treatment of bacterial infections was once at a low ebb. But since the 21st century, due to the bacterial resistance to antibiotics, has become more and more serious, bacteriophage therapy once again entered the field of vision and has achieved preliminary success in the treatment of drug-resistant bacteria and conditional pathogenic bacterial infections. 2. Principle of phage display technology and common methods. Genes encoding signal peptide of capsid protein and genes encoding capsid protein polypeptide in bacteriophages can be cut through genetic engineering technology. And then insert the gene fragments encoding exogenous polypeptides or proteins between, so that the exogenous polypeptides or protein sequences can form fusion protein molecules with phage capsid proteins to be expressed on the surface of the phage under the guidance of signals. This process is called display. In general, the preparation and screening of phages expressing specific proteins or polypeptides can be conducted according to the following steps. 1. Prepare a large number of display libraries of phage clones with a specific protein or polypeptide displayed on the surface of each phage. 2. Display libraries are used as mobile phases and target polypeptides or protein molecules as stationary phases to make the two phases contact and interact with each other. 3. Phages that cannot specifically bind to target molecule are abandoned. 4. The collected bacteriophages are used for further amplification of E. coli infection, resulting in an enrichment pool as a display library for a new round of elution.